Good afternoon, everyone. I am Obakeng Jona, as I mentioned, and from the University of Cape Town in South Africa. So the title of my talk looks at the scale-up of live biotherapeutics, in particular probiotics, for reproductive health in South Africa. So the target condition that we're looking at is bacterial vaginosis. So generally speaking, bacterial vaginosis is known as a dysbiosis of the vaginal microbiome, and it's characterized by depletion of commensal lactobacilli, which are found in the vaginal area, and it causes an increase in the acquisition of STIs and birth-related complications. And it's most prominent in Southern Africa, affecting more than 50% of females. And it's also an issue because in most cases it can be asymptomatic. And currently, antibiotics are used to treat the condition of bacterial vaginosis, and the two antibiotics that are primarily used are clindamycin and metronidazole. However, the issue with the usage of antibiotics is that in 50% of the cases after 6 months, the condition seems to relapse. That's when the research question came about, to incorporate probiotics in order to restore the vaginal microbiome. And generally speaking, probiotics are defined as live microorganisms that, when administered in adequate amounts, confer health benefits to the host. And in this instance, the host would be the female with the vagina. So the whole aim of the project would be to bring about a conjunctive means of addressing the issue with the issue of bacterial vaginosis in order to restore the vaginal microbiome. And so, generally speaking, the microorganisms that were harvested, we looked at different characteristics that the microorganisms would be able to incorporate in order to be antagonistic towards pathogens associated with bacterial vaginosis. So they need to be able to compete against the actual pathogens of choice that are related to bacterial vaginosis, as well as the production of bactericidal and virucidal compounds, such as the two enantiomers of lactic acid, as well as hydrogen peroxide. Otherwise, given the beneficial presence of these microorganisms, they need to be able to stimulate the natural host defense mechanism in the vaginal area. And generally speaking, a healthy vagina is one that's regarded to be one that is at a low pH, typically at pH levels that are less than 4.5. So in harvesting and identifying these microorganisms that are beneficial to the vaginal area, given that this project is a collaborative research project, the team of medical virologists were able to recruit 149 South African female adolescent girls between the ages of 16 and 22. And from here, 12 samples were chosen for being negative for the case of bacterial vaginosis. So in essence, it was vaginas that were regarded to be healthy that were then considered. And for these, they were then ranked for their ability to be antagonistic towards pathogens associated with bacterial vaginosis. So the properties to which I referred that are affiliated with bacterial probiotic characteristics had to do with the production of organic acids, such as lactic acid, acetic acid, or ethanol in some instances, as well as hydrogen peroxide. The microorganisms need to be able to adhere onto the ectoepithelial cells of the cervical region and to be able to inhibit the pathogens that are associated with bacterial vaginosis. And ultimately, they need to be able to lower the pH level of the vaginal area because, as I had pointed out earlier on, a healthy vagina is one that's regarded to be at a low pH. And then ultimately, given the large sample of microorganisms that were identified to be beneficial for the vaginal environment, we then ended up with selecting different strains and species, and it included Lactobacillus crispatus, Gasseri, Jensenii, as well as Leucocete as our top performing strains that had characteristics that were affiliated with being probiotic or to be able to promote the general well-being of the vaginal microorganisms. However, it's one thing to identify these beneficial microorganisms, but given that the ultimate goal of the project is to be able to scale up and to industrialize these microorganisms in order to apply them as probiotics, we then looked at the bioprocessing parameters that are affiliated or associated with these microorganisms. So the first being that they need to have a high maximum specific growth rate and cell density in order to be viable Secondly, they need to be able to reduce the pH, given that we've mentioned that a healthy vagina is one that is affiliated with the low pH, as well as the productivity of lactic acid, as well as other organic compounds that are antagonistic towards the pathogens, as well as to be able to consume the substrate. And for the context of the study, the primary carbon source was glucose. And as I pointed, the glucose was able to be converted both to biomass as well as to the antagonistic by organic compounds. We then also looked at doubling time and lag phase, because in the context of bioprocessing, these will determine how quickly one is able to produce the product, both from the lactic acid perspective or the production of the biomass. And all of this together, one is then able to look at yield coefficients, because given that they consume the product, the glucose is then converted to biomass, as is shown on the y-axis. It's converted to the product, and given these two values, you're then able to compare the extent to which the carbon source is directed toward the production of the lactic acid or the biomass. And for the focus of this project, our primary product was the biomass. So we then intended to maximize the conversion towards biomass as opposed to towards lactic acid. And given that we had all these different strains, we needed to then compare their relative scalability, because even based on the actual definition of a probiotic, it's only effective once the probiotic is in adequate amounts. So upon scaling up the microorganisms, you need to be able to have them at very high cell densities. And upon scaling up, you need to ensure that the biological activity needs to be maintained while you move from one cultivation volume to the next. So for the context of this project, initially, once the microorganisms were harvested, the studies were done in a 96-well plate by our collaborators, Happel et al., and the paper was published, and their cultivation volume was only 200 microliters. And from there, we then scaled up to shake flasks where we cultivated the 5 different strains in 100 milliliter serum bottles. This was the operating volume. And from the 100 milliliter serum bottles, we did a 3 times as well as a 5 times scale up in bioreactors as shown in this image. And then ultimately we got to 5,000 milliliter bioreactors, which was the largest volume that we had attained. And in all conditions, the growth medium in which the microorganisms were cultivated is MRS, and we cultivated them at 37 degrees Celsius, and in the case of the bioreactors, and pillar speed was 140 revolutions per minute, and in all cases, the microorganisms were cultivated anaerobically. All experiments and data were collected in triplicate, and the parameters that we looked at, we measured the OD, we recorded the pH hourly in the case of the shake flasks and bioreactors, and using HPLC, we then monitored the extent to which the glucose was consumed, the production of lactic acid, ethanol, acetic, and acetic acid. However, it's important to note that some of these microorganisms were homofermenters, and in that case, they were only producing lactic acid and not the other organic compounds. So comparing the different scales, I mean, in this first column, these are the actual growth kinetics at 100 milliliter serum bottles, and each different curve would represent a different microbial species and strain, which is shown down here at the edge. Because in blue is Lactobacillus crispatus 70.6 Pa, and it ended up being our top performer in the case of 100 milliliter serum bottles. And on the second row, you will see the actual pH, whereas in all 3 instances, the initial pH of the growth medium was approximately pH 6.5. And over time, given that the microorganisms consume the glucose and they produce organic acids, we see a reduction in the pH over time between time 0 as well as up to 26 hours of cultivation. It was also interesting to note that upon scaling up, there wasn't much of a change that was observed in the growth kinetics of the microorganisms, because if you compare the maximum ODs in the case of the 100 milliliter serum bottles, upon scaling up, it's almost as though the growth kinetics were preserved on changing the scale, although the mixing type was the same, because ultimately we had to ensure that the mass transfer as well as the heat transfer upon changing the volume of the bioreactor is the same. And across both scales, so if you look at across all 3 scales, we seemed to have a predominantly strong competitor of the top performing strain, which was the Lactobacillus aspartus 17.6, which was the top performer at 100 milliliters, 300 milliliters, as well as 500 milliliter bioreactors. So, given that this data was generated where we looked at the maximum cell density, the maximum specific growth rate, pH change, and the doubling time, alongside data that was collected from HPLC, we were then also able to look at the yield coefficient of the product, and in this instance is the proportion of the glucose that is converted to lactic acid, as well as the proportion of glucose that is converted to the biomass, and ultimately the ratio of the substrate that's converted to the biomass versus the proportion that's converted to the lactic acid. And we also then compare the rate at which the substrate is consumed and the rate at which the product is being consumed. And in all cases, you'll see that the rate of substrate consumption will be higher than the QP, because for the QP, the focus was solely on lactic acid versus the carbon from the glucose being directed toward biomass, lactic acid, as well as other organic compounds in the case of heterofermentative microorganisms. So from the previous data that we had then shown, we then chose up two top performing strains. I mean, we then went about and bought two over-the-counter strains, Lactobacillus reuteri as well as Lactobacillus rhamnosus. So these two were used as benchmarking strains in order to contextualize and compare how our microorganisms' growth kinetics compared to already established strains that are in the market. And you'll see that there's a pronounced difference between the two strains, which are in blue and red, as shown, as I point out here, compared to the novel strains that we have harvested. So these two are already commercialized, and there's a huge difference, given that they all had the same initial substrate concentration. These two strains tend to produce a much greater cell density compared to the ones that we have. But much as this is the case, from our novel strains that were harvested from healthy females in Southern Africa, we were then able to identify our top two performing strains, which was Alpha-Spartus 17.6 and L-Mucosate 19.13. These two not only were the strains that produced the highest biomass, but it was also in that their production of the virucidal as well as bactericidal compounds that are antagonistic towards bacterial vaginosis. And Lactobacillus mucosae was the only heterofermentative strains of the 5 that we had studied that we chose to have as our top performing strain. And it's important to also note and interesting to see that across all different bioreactor volumes, it seems as though the growth parameters are preserved, which then beckons the question to perhaps changing the reactor configurations in order to reach higher cell densities. And an observation also that was made, if I go back to these slides, is that upon growing the microorganisms, between the 10th and the 12th hour, we see that the pH goes below 4.5. However, if you look at HPLC data and you look at the amount of lactic acid or organic compounds that are produced, it seems as though upon going below pHs of 4.5, a certain concentration of lactic acid was reached, which speaks to two notions that it seems as though the low pH becomes inhibitory towards growth, as well as an accumulation of lactic acid within the bioreactor system becomes inhibitory to growth, because there still was enough substrates for the microorganisms to produce more biomass and lactic acid. However, the low pH and accumulation of lactic acid inhibited the actual growth of the microorganisms, which then led us to explore the usage of controlling pH. So at the same 300 milliliter working reactor volume, for the same growth medium, the MRS medium at 37 degrees Celsius, 140 RPM, and in this instance, we did duplicate runs per strain. We ended up controlling the pH because we had seen that at pHs that are below 4.5, there's an inhibition that was observed. And an interesting notion or observation was then made. In the case of Alpha-Spartus, I mean, in blue we have our control, which is the MRS. In red, it's the MRS while the pH is maintained at 6. for both instances. But then with the growth kinetics, it seems as though if you compare the red and the blue, there isn't a significant change in the amount of biomass that's reached, which speaks to the notion that although the pH is elevated at comfortable amounts, the presence of the lactic acid that would have accumulated in both instances seems to be inhibitory towards the growth of the microorganisms. And the same was done where the concentration of the nutrients was tripled and the pH was also comfortable. There was a slight improvement for the case of alpha-aspartate, but the same was not observed for L-glucoside. But again, it's the same notion that once we compare these observations to the data that was generated from HPLC, that much as the pH was maintained at 6, and in the case of the control it was dropping, once the pH goes below 4.5, the same observation was made where there was an inhibition in the growth of microorganisms. And this speaks to our hypothesis that is because of the accumulation of lactic acid or other organic compounds in the system that led to the inhibition of the growth of microorganisms not to have access to the additional concentration of substrates in the system. So in conclusion, the work was able to characterize the different lactobacilli that we were harvested, and we identified our top 2 performing strains, especially when compared to the 2 already commercialized over-the-counter strains. Secondly, we were able to identify inhibiting factors, being the low pH, as I pointed out, that pH values that are at 4.5 or less, as well as the accumulation of lactic acid in the system. Thirdly, we looked at the impact of scaling up, because across all the strains, we were able to scale up in cultivating the microorganisms at 100 milliliters all the way to 5-liter bioreactors, and we saw that there isn't a notable impact on the growth kinetics of the microorganisms. And it could also speak to the shear, because in the case of the 100 milliliter shake loss, it was being mixed versus, it was being shaken versus in the case of the larger reactors, where the impeller was actually stirring the actual reactor. However, no notable difference was seen. And with the 3 pointed out data, we were able to benchmark the growth kinetics of our novel strains against the OTC strains, and we were able to identify the actual gaps that need to be met in order to reach the high cell densities. And ultimately, we were then able to answer the question of feasibility. That much as these microorganisms have been demonstrated to be antagonistic towards pathogens associated with bacterial genesis, we then were able to demonstrate that yes, it is feasible to apply them for the case of producing probiotics in order to treat bacterial vaginosis. And that sums up the story. So this is some of the references that I have referred to. And thank you for your time. And I'm open to take questions.